All right, hello everyone, and welcome to week 7 of chemistry. Now, for those of you who have taken biology before, this slideshow might actually look a little familiar to you, and that's because it's taken from Bio 189. So the importance of this week is understanding the chemical structure and function of DNA and what it does and how it does it, more importantly. So we went over this in biology, so this is a more in-depth repeat. But I do like the biology slideshow for DNA, so that's why I decided to use it again. But talking more, of course, about the chemical aspects of what's going on here. So that's what we're going to be talking about, is DNA structure and function. And so let's get into it. So DNA and RNA synthesis here. So structure, oh goodness. So we have DNA, RNA. If you recall from our lecture on macromolecules, DNA is a macromolecule and it is made up of nucleic acids, right? That's what the NA in DNA stands for. The whole thing, of course, stands for deoxyribonucleic acid. But it's more than just nucleic acids. Those are a subunit of that. So we have the polymer, and that's the DNA, RNA, and then some of the monomers would consist of nucleotides. And then when you put a bunch of nucleotides together, you get a polynucleotide. All right, so polynucleotides can vary, but there's only 4 different types in DNA. There's adenine, cytosine, thymine, and guanine, also just known as the letters you see here in the middle of the screen, and that's A, C, T, and G. Now, those are just the nucleotides. We have to put them together, and more importantly, we have to have a structure to support them on. So we have to have what we call a sugar phosphate backbone, which in this picture here is the blue part. That's the sugar phosphate background, which of course, if you haven't guessed, it's sugar and phosphate. Now, chemically speaking, we know it's a sugar, and we know which type of sugar it is, right? Because it's deoxyribose. So ribose is the sugar, and the phosphate is in there as well. Now, getting a little more in depth here, if we just talk about the nitrogenous bases, you'll see here that there are different shapes. A is different than C and T, but very similar to G. So these are known as purines and pyrimidines. They just differ in shape and their bonding structure is different. But basically what it means is one fits into the other, kind of like a puzzle piece. So if you notice that A has this pointy structure here and T has a structure that looks like it could support that. And it's because A does indeed bond to T. And if you look at C, it's got that rounded inlet here. And then if you look at G, it's got that rounded outlet. So this will fit together like a puzzle piece. So that's the main difference between those nitrogenous bases, is their shape, and their shape will dictate whether or not they can actually bond together. But chemically speaking, let's look at this. We have a whole DNA nucleotide. So what does that consist of? It consists of a phosphate group, which is phosphorus double bonded to oxygen, and then oxygen here on top, bottom sides. It is bonded to a deoxyribose sugar. Now, deoxyribo, you might be wondering, what does deoxy mean? Well, obviously that means we have oxygen here, which is, you can see it right there on the top, and you see another one there. So it's 2 oxygen in total, deoxyribo. And then from the sugar phosphate, we get the nitrogenous base, which in this case would be thymine. And this is just an example, of course. You could also have adenine or guanine, cytosine, anything that sticks here. So just to reiterate, one complete DNA nucleotide consists of a phosphate group, a sugar, and then the nitrogenous base. That's an entire DNA nucleotide. And that's exactly what this slide says right here. It's a nitrogenous base, sugar, and a phosphate group, right? And this is the nitrogenous base right here. Now the sugar, of course, like I mentioned, is called deoxyribose, and we give the full name deoxyribonucleic acid. And of course, with nucleic referring to DNA's location in the nuclei, but we know in chemistry, nucleic acids are the types of acids that exist inside nuclei sometimes in cells that dictate, how or what a cell will do, rather. So let's talk a little bit more about these nitrogenous bases. So we have thymine and cytosine, which are single ring structures. So in other words, they look like this. See how there's single ring structures? There's only one ring here. And if we look at adenine and guanine, they are double ring structures. And again, these are known as purines and pyrimidines. So RNA and DNA polynucleotides have the same chemical structure, except that, and this is a tricky one, and I can't stress this enough, you have to know this, and it is just one of those things you just have to remember. DNA has thymine, cytosine, adenine, guanine. Right? DNA has all of that. But RNA is a little bit different, okay? RNA doesn't have thymine. It has uracil. So I'm going to say it one more time. RNA does not have thymine, that's T. It has uracil. And that's so important to remember. However, the bonding structure still remains very similar. So we know that A bonds to T, and T bonds to A in DNA. C bonds to G, G bonds to C in DNA. But in RNA, it's a little bit different. Instead of having a T, you have a U. Therefore, U bonds to A, and A bonds to U. But still, in RNA, C still bonds to G and stuff. So we'll go over that a couple of times during this lecture because it can get a little confusing. But let's take a step aside and let's talk about how we know all of this fun stuff. Well, we get Watson and Crick discovery of the double helix. This is not 100% true. Watson and Crick did do a good job. They did certainly contribute to whether or not the nucleotides were on the inside or the outside of the DNA molecule. They had a very good research plan going on, and they enlisted the help of this woman here, seen here on the right, Rosalind Franklin. And Rosalind Franklin was one of the first X-ray crystal photographers, and that is taking pictures of really tiny things using X-rays and so on. And there's a very special type of geometry that works with it. But to make a long story short, Rosalind Franklin actually took a picture, not a literal picture, but a picture using X-ray crystal photography with the DNA here. And what she found was that the DNA was not straight up and down like a ladder, nor was it just twisted once. It was what we call a double helix. It's a double helix, as you can see there. And a double helical structure means a lot of things can happen. So everything sort of fit together. Watson and Crick were pretty on the ball about the nucleotides and them bonding together, and A bonds to T and so on. They were good about that. But the actual discovery of DNA and sort of a confirmation, a visual confirmation, was done by Rosalind Franklin. So what happened was she was enlisted to do this work. She did the work. She did a great job. And basically, Watson and Crick stole her work. Now, she was mentioned in the report, but sort of as kind of like the end, oh, by the way, this lady helped us out. But yeah, she did not get the credit that she deserved. And so I wanted to point that out, that she deserves most of the credit for the double helical model. Okay, so this slide just talked about basically the same thing here. So let's talk about DNA replication. Now, in each one of your 75 trillion cells, the DNA in those cells has to replicate somehow in some way. There are a few exceptions to that rule, so it's pretty common to find. So when a cell reproduces, it has to make DNA for its other cell, right? And Watson and Crick's model of DNA suggests that each DNA strand serves as sort of a mold or a template to guide reproduction of the other strand. In other words, the double helix model allows for replication of the DNA. So DNA inside a nuclei is going to split up. And it's going to do this with various enzymes. But the enzyme I want you to focus on right now is DNA polymerase. Now, you know it's an enzyme because it ends with an ACE, right? A-S-E. So DNA polymerases, that's plural, those are enzymes that make the covalent bonds between the nucleotides of the DNA strand and can help repair DNA that has been damaged by toxic chemicals. And to be clear here, there are hydrogen bonds between the actual nucleotides and not covalent bonds. Pardon me. So DNA replication begins on a double helix at specific sites. And we know this as the origin of replication because DNA, as it turns out, is extremely long, and one chromosome holds a ton of information, most of which we don't really know what it does. So it's not like one DNA polymerase comes along and copies all 23 pairs of your chromosomes. That's not it at all. In fact, it takes thousands upon thousands of these enzymes per cell just to, replicate DNA once. So instead of looking at DNA like, okay, well, we're going to start here at the left and replicate to the right, it actually starts at two different sites of origin, and they sort of expand out. So the orange right here is what we call the daughter DNA. It's the new DNA that's being made from this mold. And then they'll, of course, meet here in the middle, and then you get two different strands of DNA. So the origins of replication, there are multiple of them. And that's because it would take forever if it was up to just one DNA polymerase. So DNA replication serves a lot of purposes. One being, of course, that it ensures that all cells in the body carry the same genetic information. Is it perfect? No, it's not perfect at all, but it is essential because for that very reason I just read. It also means that genetic information is passed to offspring. This is more prevalent, obviously, in meiosis, making gametes, sex cells, sperm and egg cells. It's semi-conservative, which of course means that DNA replication produces two daughter DNA molecules with each one strand from the parent DNA. It's semi-conservative. So in other words, you have the parent DNA and you get two different sets of DNA afterwards. Now, we'll talk a little bit more about this later, but for right now, DNA is the recipe book to make you who you are. And what do you need? Think about a recipe book at home. What do you need? You need there to be maybe it's a book, maybe it's a binder, maybe it's a folder, it could be a lot of things. But you need the recipe book to be in a language you understand, right? Like I couldn't take out a recipe book in French and make it perfectly. No, I would need a translator. Right? I would need something to help me translate that information. And that's kind of what your body does. Your DNA speaks a different language than your body's used to. So we have to go through these different processes to make it the same language so that we can make whatever we want to make. And this is known as the central dogma of biology, going from DNA to protein, because it is the driving force behind everything biology, which is why it's the central dogma. And this creates two different things. And for the focuses of chemistry, I don't really focus on this too much. This is more genetics, but I wanted to just get this out of here. On the bottom, it says genotype and phenotype. So a genotype is when you have the genes, right? When you have the trait, you carry the trait in your genetic material. The phenotype is the physical trait that you have. For example, I have blonde hair and blue eyes. My family has mostly blonde hair and blue eyes, except for one of my sisters who has red hair. And that means her phenotype is red hair, but she most likely has a genotype somewhere that might code for blonde hair. Or I, on the other hand, might have a genotype that codes for red hair. So my genotype might be red hair, but my phenotype is blonde hair. And this, the reason why this happens, of course, is because of dominant and recessive alleles. So some hair color is more dominant than others. Like blonde is more dominant than red, and black and brown are certainly more dominant than blonde. But that's not the focus of this class, but I did want to talk about the differences between the two. This is a slide that repeats itself a couple times, so I just want to make sure that we get past it. So remember, DNA is the recipe book. We got to transcribe what it says, and then translate what it says. So there's a lot of different ways to figure this out, but I like the recipe book way the best. Those of you who are musicians and who can play or can read music, maybe you can sight read, you know what transcription and translation is from a musical aspect. When you transcribe music and you can translate music. But anyways, so DNA in the nucleus is unreadable. We can't understand what it's saying, but it's telling us something really important. It could even be life or death. So we transcribe the information that the DNA is saying, and we send it off to the ribosome, and the ribosome translates the information into a message that we now understand. So that's the step. We go from transcription to translation. Now, transcription always happens in the nucleus. Translation always happens in the cytoplasm. So what we're making here is protein. That's the main thing that we're making. We're not making sugars. We're not making fats. We're making protein. And think about it. We talked about this in an earlier class. What are proteins made up of? Amino acids. So if we can make a bunch of amino acids, then we can make protein. So the language of DNA is actually coding for protein indirectly, but it is still coding for protein. So we have these little codes right here. You see this A, C, C, G, G, A, and so on. You know, look at their counterpart here. Instead of, because this is an RNA strand, instead of T, it has U's. You'll have U, G, G, C, C, G, U. Okay. Each codon, and when I say codon, a codon is 1, 2, 3 nucleotides. For every 3 nucleotides, you get 1 amino acid. And those 3 nucleotides are called a codon. Therefore, for every 1 codon, you get 1 amino acid. It's important that you know that. So a codon consists of 3 nucleotides, and 3 nucleotides will code for 1 amino acid, which means that when your DNA, or rather the ribosome, is reading the mRNA strand, it's reading it in groups of 3. So it's reading it every 3 nucleotides. And because of this, every 3 nucleotides will have different amino acids. So the order in which those amino acids are put into place will determine the protein that is made. So this is known as your genetic code. So the genetic code, the thing that makes you who you are, is basically a sequence of amino acids at the very end of translation put together to make protein. Now, humans share, 99% or more than 99% of their DNA with each other, but there is a very, very subtle amount of DNA that's not being shared, which is the part of the genetic code that makes you unique to yourself and the rest of your species. So the genetic code is a set of, not rules, but it's a set of codes in your DNA that make you who you are. And it also makes you a human being and not another animal. And so the codes can be pretty tricky to read, but this slide right here is a pretty good indication of, a pretty good indication of how many codes can code for one thing. So for example, if we had, let's see here, if we had an RNA codon that began with, that would just said CUU, well, that would be this amino acid. If we had CCU, it would be proline, or PRO. If we had GGU down here, it would be glycine. So these are codons, because remember codons are 3 amino acids right here. These are codons, and those are going to code for these amino acids, and that's what you're seeing in here, right? So these are all amino acids. Now, if you notice that it says start and stop, and these codons are probably the most important ones that we have, because AUG is methionine, and that will start the process of translation. And then these 3 codons here, UAA, UAG, UGA, will be your stop codons. Now, to be clear, I do not want you to go home and remember every single code. Please don't do that. Not even the stop or start codons. Just remember that every codon codes for 1 amino acid. So RNA follows some different rules than DNA. So let's just kind of take a step back and then we'll move forward here. When DNA wanted to replicate itself, what did it use? It used DNA polymerase, right? DNA polymerase makes DNA. That is short and sweet, and there you go. That's the long and short of it. When you want to make RNA, you use RNA polymerase. Hopefully that makes sense. So DNA, to make more DNA, you get DNA polymerase. If to make more RNA, you make RNA polymerase. Rather, you use it. So here's the DNA right here. Just here it is. RNA polymerase is going to come along. It's going to unzip the DNA, and it's going to start making what we call mRNA, and that'll be on the next page, right here. I'll come back to the slide in a second. So this mRNA is made from RNA polymerase. So RNA polymerase is going to, latch on there and make mRNA. The M, of course, stands for messenger RNA. And this is all transcription. This happens in the nucleus. Now, there are some parts of mRNA that we don't need, right? So first off, as soon as mRNA is done doing its job, and it's completely made and it's single-stranded and all that fun stuff, it's going to get a cap and tail, right, to kind of validate it. But there are some parts of that mRNA that we just don't need. And we call these introns and exons. Introns, exons right here in RNA splicing. So inside the nucleus, the mRNA is going to be sort of edited, because there are some things we need, some things we don't. So introns are non-coding regions, meaning they don't do anything, so we don't need them. Exons are coding regions. These are the genes. These are the things that we want to be read. So here's how I used to remember it. Introns stay in the nucleus. Exons exit the nucleus. And you can see in this picture that it certainly does exit the nucleus here. Introns stay in the nucleus. Exons exit the nucleus. So let me go back to this one, 3-phase of transcription. So you have the initiation phase, which is the attachment of the RNA polymerase to the promoter. And the promoter, by the way, is just a region of DNA that says, hey, start here, And then the elongation phase, where you start to make more mRNA molecules. And by the way, mRNA is a single-stranded, it's not double helixed or anything. And then finally, the termination phase, where it gets cut off, and then you're done. So initiation, elongation, termination. Those are the 3 phases of making mRNA. Now, the 3 major players here, we've already mentioned mRNAs, but I like to move on to the rest. We have tRNA, which stands for transfer RNA. And these guys are very closely shaped like a T, but they have the amino acids. So the amino acids just don't make themselves. They are actually, 10 of them are made by themselves. Most of the amino acids that you're going to get through your diet, and then of course the rest are being made. It is half and half though. You get 10 amino acids that you make, and then 10 that you get from your diet. But regardless, amino acids exist in your cells already, and they attach to these things called transfer RNAs. So they're holding the amino acid, and they're going to become very important in a little bit. And then you get ribosomal RNA, or rRNA, lowercase r, rRNA. And this just makes up ribosomes. That's what ribosomes are made of. They're made up of rRNA. So let's take a look at tRNA a little bit closely. So again, tRNA is a molecule that's made up of single-stranded RNA, one polynucleotide chain consisting of 80 nucleotides total, because RNA is nucleic acid, so it has nucleotides. The chain twists and folds itself, forming almost like a T shape. Now, what's really important for chemistry, what you need to know here, is the transfer RNA, the tRNA, contains what we call a codon, sorry, an anticodon. So for every codon, you have an anticodon. Now, anticodon doesn't mean it's against or it's gonna like cancel it out. I mean, sure, in a sense, yeah, but it's not anti-anything, it's nothing bad. Anticodon just means that it's gonna bond to a codon, that's all it is. So we use this during translation, right? Not transcription anymore, because we did transcription, we're done. Translation happens in the nucleus. Oh, no, it happens in the cytoplasm of the ribosome. And those anticodons will hook up with those codons that are on the mRNA strand. And if this is making no sense, don't worry, I have a video to smooth it out. So, and again, ribosomes are made up of rRNA, and they're the ones that are actually going to read it. Now let's put all of this together. Yeah, here we go. Here we have all this blue stuff here. This is a ribosome, and a ribosome comes in 2 units. It has a large subunit and a small subunit, and this is made up of all rRNA, right? Ribosomal RNA. Now, mRNA is going to come from the nucleus, and it's going to bond to the mRNA bonding site right here. So this everything in red is the mRNA, and then these tRNAs, they're floating around the cytoplasm, and they're going to come down and they're going to hook up with the correct codons because they have to bond to each other correctly. Now, every time they do that, you see here, you see this amino acid right here. Every time they do that, there's a peptide bond that forms between these 2 amino acids, and the protein chain gets longer and longer and longer. I should say the amino acid chain gets longer and longer. So that is basically the end of transcription. Pardon me. So what I'd like you to do is just to go back for a second and review that, because that's pretty difficult. I'm going to pause, and then I'm going to have a video to watch in just a second. Okay, and I'm back. So this is a video that I've watched a million times. It's a 2-minute, 41-second video. It's very short. I am going to turn the volume off on it though, but I do want to show you all the steps of DNA transcription and translation. So let's take a look here. So we have, of course, an animal cell, and we're about to zoom in side of it. So we're going to see all of the organelles and all that fun stuff, but we're really focused on the nucleus, which is right there. We're going to pass through a nuclear pore right about now. And here we have the chromosomes. Now, chromosomes are just wound up pieces of DNA. So all we're going to do is we're going to unwind the chromosomes, and there's all the DNA wound up around those histones. Now, we only want one section of DNA, and this is called a gene. So we're going to get RNA polymerase to come around because we want to make mRNA. So RNA polymerase is going to come around. There it is. RNA polymerase is going to attach, and then it's going to run through, and it's going to bond everything just as we talked about. A's, T to A's, U to A's, G to C's, and so on and so forth. It keeps doing this over and over and over again. Remember, this is the elongation phase. And once it's finished, that's the termination phase. And here we have a finished messenger RNA, or an mRNA strand. But we don't need all of it, right? We're going to cut out pieces that we don't need very soon. There we go. We don't need that piece, so we're going to cut it out, splice those things together. Now we have an exons, right? Exons are going to exit the nucleus just like this. It's going to move to the outside. It's going to hook up with a ribosome. And remember, ribosomes come in two pieces, and they're made of rRNA. And so here comes the ribosome, clamps down on it pretty good. And now the ribosome is attached to the messenger RNA, but then we get this guy right here. This is the tRNA. tRNA has the amino acid on the top and then the anticodon on the bottom. And the anticodon is responsible. See how the anticodon has the letters? It's responsible for bonding to the correct codon. So I'm going to pause the video. So remember, this is the mRNA strand. These are the anticodons. Take a look here. C bonds to G, U bonds to A, C bonds to G, U bonds to A, and so on and so forth until the end. So it's the same principle as what we've been talking about. So when you go from mRNA, if you want to bond mRNA to tRNA, it's the same rules as before. So I'm going to continue. So that it's going to keep going on and on and on. And then every time we do this, those amino acids are bonding via peptide bonds, and they're getting longer and longer and longer and longer for each time. And then eventually we're going to get what we call a simple or primary structure of protein. So that primary structure of protein just detached. It's going to fold in secondary, tertiary, and then quaternary structure. And then we get a protein. Now this protein here, probably, it could be an enzyme. It could be a hormone. It could be part of the cell. It could be keratin. There's a million uses for protein, and which is why your body uses it quite often. So I encourage you highly to go back and review that video, and with me, of course, describing it to you. Now, if you want to look at it for yourself, all you got to do is visit, of course, YouTube and just type in DNA transcription translation. And the actual name of the video is From DNA to Protein 3D. That's the actual name of the video. Highly recommend you go over this as many times as possible, because it's really going to help you a lot. You can see here it's got over 7 million views. I'm probably at least half of those views, because I do use this all the time. Okay, so if you like, I suggest you pause here again and review that chapter, because we're about to get into another chapter. Okay, and here we are in chapter 6. So chapter 6 is going to be talking about cellular respiration and metabolism, which is another part of this course that we need to learn about. So in your body, the number one thing that uses most of the energy that you consume is your muscles. Your muscles need continuous amounts of energy to keep you breathing, to keep your heart going, to keep you digesting, keep you sitting up straight, standing up straight, and even lying down to some degree. Now, your aerobic capacity is the amount of oxygen in which you can take in. And there are some processes in your body that do not require oxygen, but they have sort of a downside to that. So I'm going to try to make this as quick and painless as I can. We're not going to talk about photosynthesis, autotrophs, or heterotrophs right now, since this is for biology. But I wanted to talk about the circle of energy, essentially. So I want to focus on, hold on a second. I want to focus on this picture. This picture it doesn't seem very important, but it really is. So I want you to think about this for a second. So the sun, that's 93 million miles away and is basically a nuclear fusion factory, makes these rays, these sun rays that come down, the solar UV rays that come down, and they provide energy to our plants, trees and bushes and grass and so on. Okay, so the sun beats down on these plants, and these plants take that energy, kind of like solar cells, and they make sugar, that's C6H12O6, and oxygen. Now, guess what, guys? We need sugar and oxygen to survive. You and me and all animals need sugar and oxygen to survive. And that gets digested in the mitochondria. And that's so we're gonna break down that sugar and we're gonna use oxygen as a final electron acceptor. Now, when we do this, we produce water and CO2. Well, guess what? The plants need the water and CO2 to make the sugar and oxygen for us so that we can make water and CO2 for them and so that they can do this. So it is this circle of life here, the circle of energy. We feed the plants and they feed us. It's been like this for a long time. So as a matter of fact, the photosynthesis and cellular respiration chemical formulas are opposite each other because of this very reason. So cellular respiration is the aerobic harvesting. That means taking oxygen from the air. And if it's an aerobic process, that means it requires oxygen. If it's anaerobic, it means that it does not require oxygen. And as a matter of fact, it's anaerobic would mean, sometimes could mean that it could be poisonous, especially for some bacteria. So it requires that all cells exchange two gases with its surrounding. That would be CO2 and O2. Now, this here on the bottom is the chemical formula, or sorry, the chemical equation for cellular respiration. So what do we get? C6H12O6, that's glucose, plus 6 oxygen will yield 6 carbon dioxide and 6 water, plus approximately 32 ATPs. Now it's between 32 and 36. You know, some scientists speculate more or less, but that's not the point. The point is we take in we take in oxygen and sugar, and we give out carbon dioxide, and we give out water. Now there are 3 steps to this. There are 3 main steps that we do to go from sugar all the way to carbon dioxide and water. Carbon dioxide comes out pretty quickly. Water takes a little bit. So there are the 3 main steps. We have glycolysis. Glyco would refer to sugar. Lysis means to destroy. So this is literally destroying sugar. The citric acid cycle, also known as the Krebs cycle, and I'm going to talk about it using Krebs and citric acid cycle both. And then finally, the electron transport chain, which is the ETC. Now, during glycolysis, a molecule glucose, that's C6H12O6, is split into 2 molecules of a compound we call pyruvic acid, or pyruvate for short. The citric acid cycle, also called the Krebs, uses enzymes that make CO2, and they also get ready for the final stage, which would be the electron transport chain, which is this stage right here. So during glycolysis, located generally in the cytoplasm, so this doesn't happen in the mitochondria, this happens outside, you're going to get glucose, right? Here we go, we got glucose, and in order to get the process started, we actually need to contribute 2 ATP. So we're going to do that. We're going to contribute 2 ATP, and we're going to get 4 ATP in return. And now it doesn't say it on this slide, but it does say it on the next. We'll get 4 ATP in return, which means we have a net gain of 2 ATP. And I'll explain it in dollars and cents. So if you give someone $2 and they give you 4, you just made $2. It's essentially what's happening here with ATP. We give 2 ATP to get in, but we get 4 once we're in, and so we net 2 ATP. Now, in addition here, we also have 2 NADHs. This is really important. So NAD is an electron or hydrogen acceptor. This is known as redox, oxidation reduction reactions. And we're going to get NAD. We're going to go from NAD to NADH. The H, of course, stands for hydrogen. So NAD to NADH. Now, from here, the pyruvic acid has to be groomed, in other words. So we're going to transform it from 2-pyruvate to acetyl coenzyme A, or you could say just acetyl CoA if you want to. Now, when you do that, you actually give off CO2 right away. So there's no ATP that's made from this part, but we do get an NADH out of it. But to go forward, the Krebs cycle, we have the pyruvic acid, which turns into acetyl coenzyme A, and then it goes through the citric acid cycle twice. So this turns twice, and we get a total of 6 carbon dioxides that get released. So there's our 6 carbon dioxides. That's where you get in the citric acid cycle. Net energy transformation for both pyruvic acid is 8 NADHs plus 2 FADH2s. These are, like I said, electron shuttles. And we get a net total of 2 ATP. So the essentials for the citric acid cycle would be that we go from pyruvic acid, which we got in glycolysis, to acetyl coenzyme A. Acetyl coenzyme A is the real beginning of the citric acid cycle. Now it goes through this twice. We release 6 carbon dioxides. We get 8 NADHs and 2 FADH2s, and then 2 ATP. And then I would like to speak really quick about these NADs and NADHs, because I feel like they get kind of glossed over. What are we doing when we're getting all this hydrogen? Well, hydrogen, as you all know, has an atomic number of 1, which means it has 1 proton, and it can have 1 electron as well. Now, hydrogen doesn't really like its electron too much. They have a very love-hate relationship. But through the transfer of this 1 electron, we're going to have a ton of energy pushed out later on. So your cell, your mitochondria, to be more specific, is very smart in that it collects all of these hydrogens to be used later. And the hydrogens that will be used later will be used in the electron transport chain. So look at this. Don't read the top here. Look at the picture. You see how we go from NADH to NAD? We got rid of the H. Well, where does that H go? Well, look, it gets pumped out into the inner membrane of the mitochondria. It gets pumped out, a couple of times, and then you have all of this hydrogen being pumped out. This is a lot of energy. And then you increase the concentration of hydrogen here, it's going to want to go back in, because high to low is where things will go. Now, when all of that energy is passed through this enzyme called ATP synthase, you're going to make ATP, and a lot of it. So the electron transport chain makes the majority of ATP in your cells. Now, what happens here? Well, we have a lot of hydrogen coming back, and I really haven't even mentioned oxygen, and you breathe oxygen all the time. So what's the point of breathing oxygen if we don't even use it? Well, here it is, guys, right here. Oxygen, and when it says half of O2, it just means 1 oxygen atom, binds itself with 2 hydrogen to make H2O, water. So the only reason you breathe oxygen is to use this at the very last step to collect all the hydrogen, and then that's it. I know breathing is important, don't stop, but all of this reason for breathing is just to collect oxygen so that we can make water at the very, very last step. So we should probably review that, right? So let's review this again. So glycolysis is your first step. You're going to start with glucose. You're going to get 2 ATP out of it. It doesn't require oxygen, and you make 2 pyruvic acids, sometimes just called 2-pyruvate. In addition, you're also going to make 2 NADHs, and we're getting ready to go to the ETC. From there, we're going to go to the citric acid cycle, but in order to get there, we have to go from pyruvic acid to acetyl coenzyme A. And acetyl coenzyme A is going to cycle through the Krebs cycle twice. And we're going to get 2 FADH2s, 6 NADHs, 6 carbon dioxide, and of course 2 ATP. And this does not require oxygen right now. There are some parts that do, but not for the focus of this class. No, it doesn't. And the most important things that we've made so far is NADHs, 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 and then FADH2s. And then all of those H's, all of those electrons essentially get pushed down, and we get about 28 ATP. Now, it's between 28 and 32. I know that for sure. Uh, I think the standard is about 36 ATP in total. That is with glycolysis, Krebs, and everything else. So just side note there. But about the majority of your ATP is made in the electron transport chain, which is done by pushing those NADHs, or rather the H's, out, and then creating a membrane potential, and then passing them through ATP synthase and making 28 ATP. So as I mentioned before, your body sort of has a hierarchy of what it prefers. If carbohydrates are available, then it'll go through the very same process that we just talked about, right? Glycolysis, Krebs, etc. Done. But let's say you run out of carbohydrates. Now patients who choose to do the what's it called, the keto diet, that's what they're doing. They're not eating sugars, but they're eating a lot of fats. And the process of burning fat or converting to burn fat would be called ketosis, right? And so this was actually happening on a chemical level. So instead of having carbohydrates to burn, your body's now going to burn fat. Well, in order to do this, it has to break it down into two different parts: glycerol and fatty acids, which is exactly what lipids are made out of. So glycerols and fatty acids. The glycerol goes to glycolysis, and it's going to be torn apart to make 2-pyruvate. Everything is the same. The fatty acids actually go and help make acetyl coenzyme A. That's the only difference there. And finally, and most complicatedly, let's say you burn all the carbohydrates, you've burned all the fat, all that's left in your body is proteins. Your body doesn't get a whole lot of energy from proteins, but it'll take what it can get. And we see this a lot in patients who are anorexic and bulimic. We'll see this a lot because they've burned through all of their energy reserves. And so there's nothing left but their body. So their cells literally start to eat the body from the inside out, which is why a lot of patients look like they're skin and bones. So proteins are, of course, made up of will break down to amino acids. And these amino acids will play various roles in glycolysis, Krebs, and of course, making acetyl coenzyme A. They're not very efficient, but they will eventually get the job done. But they will all lead to the electron transport chain. So you could go without eating for a long time, as long as you can burn all of these, macromolecules. Now, water is different, right? You can't go without drinking water. Even though we make water at the end of it, doesn't mean that you shouldn't drink it. We can only go about 3 days without water before we dehydrate. But these three, you can go about 3 weeks to a month, but it depends on the person, mainly. Now, what happens when you don't have any oxygen, or not enough oxygen, I should say? Well, patients who maybe don't exercise too often and they decide to sprint really quickly will go through a process called anaerobic fermentation, which means without oxygen. And so basically it's just glycolysis. We just talked about that. It's just glycolysis, which doesn't require oxygen, but it's only going to make 2 ATPs, and that's it. The downside, a huge downside for this, is that humans, when they do this, they create something called lactic acid. And lactic acid is a product that gives you muscle cramps and muscle strains and all that stuff. And if you've ever been sitting down for a while and then decide to get up really quick, you might know what a muscle cramp is. And what do you do to the area? You massage the area to make the pain go away. Well, why does that work? Well, when you massage an area, you actually stimulate more blood to come to that area, and that blood sort of washes that lactic acid away, and therefore the pain will go away. Now, in addition, using bacteria, we can do what we call alcoholic fermentation. It's ethanol fermentation specifically. It's been done for a very long period of time. As we know, ethanol contains 2 carbon atoms in it because it's an alcohol. And it's just a byproduct of glycolysis that happens in anaerobic bacteria. And they're in charge of making beer and wine and all that fun stuff. So I highly, highly, highly recommend you go through this multiple times, because both subjects that we talked about today were on the tricky side, right? They were not easy. So I highly recommend you go through and look at DNA replication, transcription, translation, and then go into your glycolysis, Krebs, and etc. I don't have a video to show you about this. I feel like it just sort of adds to the confusion, but you're welcome to look up a few. And yes, so please let me know if you have any questions. I understand this can be very difficult, but again, I highly encourage you to, to go through this a couple times. Now, on a final note, there's a few things because next week is our final week. I want to make sure that you have all of your quizzes in. Any quizzes that are incomplete will be given a 0. So make sure that gets done. Any missing Labster assignments or lab assignments will also be given a 0 as well. Your lab practical will be an average between all of your Labster grades, right? So if you don't do a Labster grade and it becomes a 0, well, then that 0 is going to be included in the average and your lab practical grade will go down. So keep that in mind. So yeah, chemistry quizzes, Labster, and that's pretty much it. If you got those 2 things, you're good to go. Now, if you're having issues with Labster, it's important to communicate with me. Again, not the last week, because it's going to take time for me to get back to you. The sooner, the better. So anyways, guys, if you have any questions, please let me know. If not, I will see you all next week. Take care. Bye-bye.